An invisible war is being waged across the globe. It's a conflict fought in dense jungles, remote savannas, and deep oceans. On one side are sophisticated, ruthless criminal networks that treat our planet's most iconic species as mere commodities. On the other is a dedicated and increasingly high-tech alliance of scientists and conservationists. The illegal wildlife trade, a multi-billion-dollar transnational crime, is pushing thousands of species towards extinction. For years, traffickers operated with impunity, their complex supply chains obscuring the link between a carved trinket in a market and a slaughtered elephant thousands of miles away. But the tide is turning, and the key to this disruption lies within the very blueprint of life itself: DNA. This is the story of how DNA is rewriting the rules in the war against wildlife crime. To appreciate the disruptive power of DNA forensics, you first have to understand the enemy. Wildlife trafficking isn't the work of lone, opportunistic poachers. It is a highly organized, multi-layered criminal enterprise, ranking among the most lucrative illicit trades in the world, on par with the trafficking of drugs, arms, and human beings. Estimates of its annual value range from $7 billion to a staggering $23 billion, money that finances corruption, funds armed militias, and destabilizes entire regions. The structure of these trafficking networks is like a pyramid, designed for maximum profit and minimum risk. At the base are the poachers, the individuals who carry out the physical act of killing animals or illegally harvesting plants. Often from impoverished local communities, they are the most visible and most easily replaced cog in the machine, earning the least amount of money. The next level consists of local brokers who purchase the raw products, ivory, rhino horn, pangolin scales, from the poachers, consolidating the goods for onward sale. Above them are the national-level intermediaries. They are a critical link, consolidating even larger quantities of wildlife products and laundering them into what may appear to be legitimate supply chains. Corruption is rampant at this stage, with officials often bribed to create fraudulent permits or simply look the other way. Then come the architects of the transnational movement, the exporters and importers. Based in major cities with access to international ports, they are experts in concealment and document forgery. They exploit legitimate transport systems hiding ivory inside shipments of agricultural products, concealing pangolin scales in containers of scrap metal, or using express mail for smaller items. Their routes are constantly changing to evade law enforcement. Once the shipment arrives in a destination country, predominantly in East and Southeast Asia, wholesalers take over. They process the raw materials into finished goods and distribute them to retailers. These products are then sold in physical markets or, increasingly, through online platforms that offer sellers anonymity and a global reach. And at the very apex of this pyramid are the consumers. Their demand, driven by status, tradition, or perceived medicinal value, is the ultimate engine of the entire trade. A piece of carved ivory is a luxury good. A rhino horn is wrongly believed to cure diseases. Without this demand, the entire criminal structure would collapse. For decades, identifying this contraband was a frustrating task for law enforcement. Once an animal is processed, a tusk carved, a horn ground into powder, a hide tanned into leather, visual identification becomes nearly impossible. This ambiguity was a get-out-of-jail-free card for traffickers, but DNA analysis has shattered this barrier, offering a suite of techniques to answer the critical questions of any investigation. What is it? Where did it come from? And who is responsible? The most fundamental tool is species identification through a method called DNA barcoding. Think of it like a universal product code for every species. Scientists focus on a short, standardized section of DNA, typically from a gene called cytochrome C oxidase I, or COI. This gene region is consistent within a species, but highly variable between different species. The process is elegant. Scientists extract DNA from a confiscated sample, a sliver of meat, dust from a horn, a leather-like pangolin scale. They amplify the barcode region and read its genetic sequence. This sequence is then compared to a massive reference database containing millions of barcodes from verified species. A match provides a statistically certain identification. This technique is a game changer. It's been used to identify cooked meat from a poaching case as an endangered deer, to confirm that shark fins sold in markets come from protected species, and to analyze traditional medicine pills and find the DNA of snow leopards. Identifying the species is just the first step. To dismantle trafficking networks, investigators need to know where the animal was poached. This is where population genetics comes in, using genetic markers like single nucleotide polymorphisms, or SNPs. These are tiny single base changes in a DNA sequence that create a unique genetic signature for different populations of the same species. By sampling DNA from animals in known locations, often from dung collected non-invasively, scientists can build a reference map that links these SNP signatures to specific geographic areas. When a new, unknown sample is seized, its SNP profile can be compared to this map to infer its most likely origin. A landmark study led by Dr. Samuel Wasser at the University of Washington used this very technique on large ivory seizures. By creating a comprehensive SNP reference map from elephant dung collected across Africa, his team was able to trace the majority of large ivory shipments over a decade back to just 2 poaching hotspots. This proved that ivory from a seizure in Malaysia could be traced to a specific protected area in Tanzania or Zambia, directly linking a consumer country to a crime scene thousands of miles away. Another powerful marker is the short tandem repeat, or STR. These are short, repeated sequences of DNA, and the number of repeats varies highly among individuals. This is the same technique used for human DNA fingerprinting at crime scenes. In wildlife forensics, it allows scientists to create a unique genetic profile for an individual animal. A seized rhino horn can be matched directly to the carcass of a poached rhino, providing an undeniable link between the trafficker and a specific poaching incident. This elevates the crime from simple possession to poaching, enabling much harsher penalties. This concept is the basis for the Rhinoceros DNA Index System, or RODIS, a database that stores the unique STR profiles of thousands of rhinos. When a horn is confiscated anywhere in the world, its profile can be run against the database, linking it directly to a crime. The theoretical power of these tools is made real in their application. Take the case of the ivory cartels. For years, authorities seized massive shipments but couldn't connect the dots. Dr. Wasser's DNA analysis changed everything. By matching tusks from the same individual elephant, or from close relatives like parents and siblings found in different shipments, they proved that these seemingly isolated incidents were the work of the same trafficking networks. The DNA evidence provided a roadmap that shifted the focus of law enforcement from low-level poachers to the international kingpins coordinating the trade. This technology isn't limited to animals. The illegal logging trade is just as destructive, but identifying timber is notoriously difficult. In one case in the United States, law enforcement suspected a timber mill was processing illegally felled big-leaf maple trees from a national forest. Investigators collected wood samples from the poached stumps in the forest and from processed wood at the mill. Using DNA fingerprinting, forensic scientists were able to match the timber in the mill directly to the stumps. The genetic match was incontrovertible proof that led to successful convictions, sending a powerful message that traffickers could no longer hide behind the anonymity of processed wood. And consider the pangolin, the most trafficked mammal in the world. Seizures often involve tons of scales from thousands of animals. In April 2019, Singapore authorities intercepted a record 13 metric ton shipment of pangolin scales mixed with ivory. DNA barcoding identified 3 different African pangolin species among the scales. But more importantly, the fact that over 25% of large pangolin scale seizures are commingled with African elephant ivory, confirmed by DNA, strongly suggests that the same major criminal syndicates are trafficking both, using the same established routes. Despite its transformative potential, the path from a DNA sample to a conviction is not always smooth. The biggest challenge is the database deficit. The power of any DNA matching technique depends entirely on the reference database. While incredible progress has been made for elephants and rhinos, vast data deserts exist for thousands of other trafficked species. Building these databases is a monumental and expensive task. Another hurdle is legal admissibility. For DNA evidence to be used in court, it must be collected, handled, and analyzed according to strict, validated protocols. The wildlife forensic community is still working to establish the kind of internationally recognized standards that exist for human forensics. Finally, there's the issue of cost and capacity. Building and running a wildlife forensic lab requires significant investment. Many of the countries richest in biodiversity are also the ones with the fewest resources to dedicate to this high-tech fight. But the field is evolving as rapidly as the trafficking networks it seeks to dismantle. One of the most exciting developments is the advent of portable DNA sequencing technology. Devices like the Oxford Nanopore Minion, a sequencer the size of a USB stick, are bringing the lab to the field. This,